Hello and welcome. I'm Tom Fabian, Clinical Education and Science Consultant with Diagnostic Solutions Laboratory. In this video, I'm going to be talking about the new GI map microbial markers, and especially focusing on what clinicians need to know to better understand these new markers, how to interpret them, and then how to work with them in practice. This next slide shows the new microbial markers that have been added to GI map. And these are the markers that I'll be discussing in this video. And they are Fusobacterium prausnitzii, Akkermansia muciniphila, Methanobacteriaceae, and Fusobacterium. So before we dive in, I just want to give a quick background on why we decided to add these particular markers to GI map. Overall, the goal is to continue to enhance the clinical applicability of GI map, particularly based on the latest research. And then taking this research into account, we developed several different criteria for helping us to determine which microbial markers were most appropriate for inclusion in the new version of GI map. And that included, in particular, identifying what are called keystone species. And these are species that are known to be especially important to the overall functioning of the gut microbiome. Markers were also selected based on their ability to provide clinicians with additional insights into the health of their patients, in particular in the areas that GI map already has a very strong focus in, which is in digestive function as well as immune function. And then lastly, markers were also selected based on their ability to be modified in practice by known clinical approaches. So that gives you a little bit of insight into how the new microbial markers were identified and selected for inclusion in the new version of GI map. So let's go ahead and look at the new version itself in terms of the GI map report. And so the new markers, the 4 new markers, are located in 2 sections of GI map. So the first 2 are located in the normal bacterial flora section, and they are Akkermansia muciniphila and Fusobacterium prausnitzii. And then the other 2 markers have been added to the opportunistic bacteria section. The first of those 2 markers is the Methanobacteriaceae family, which is listed in the additional dysbiotic and overgrowth bacteria subsection. And then the next marker, the 4th marker, is Fusobacterium, and that has been added to the potential autoimmune trigger section. So again, these are the 4 new microbial markers that have been added to GI map. And in the rest of this video, I'm going to be talking about each of these individual markers and telling you a little bit about each of the markers, what's known in terms of their characteristics and their functions in the microbiome, as well as how they are related to health and disease, and then potentially what's known about modifying them in practice. So let's go ahead and start off with the first marker, which is Fusobacterium prausnitzii. And again, that has been added to the normal bacterial flora section. So these are some of the key characteristics of Fusobacterium prausnitzii. It's well known as a keystone species, and again, that means it's known to play an especially important role in the overall function of the gut microbiome. It's also known to be a major producer of the short-chain fatty acid butyrate, and butyrate itself has many well-established roles in promoting health. Fusobacterium prausnitzii is also one of the most abundant species in the large intestine, and it has a prominent role, as I mentioned, in the gut ecosystem, not only in terms of promoting or producing butyrate, but also produces other factors that help support the gut ecosystem. Most importantly, though, with regard to health, it's known to promote a healthy intestinal barrier, as well as anti-inflammatory immune responses, particularly by stimulating what are known as regulatory T cells. All right, so this next slide summarizes some of what's known about associations between imbalances in Fusobacterium and relationship for various diseases. Most of those associations with regard to Fusobacterium have to do with relatively low levels in individuals that have various inflammatory and autoimmune conditions. And those conditions include things like inflammatory bowel disease, diabetes, cardiovascular disease, and colorectal cancer. And this is something that's been pretty firmly established in many different studies, as well as meta-studies, meta-analyses, where they're looking at a wide range of conditions, and by and large, in many of those conditions, Faecalibacterium prausnitzii tends to be lower than healthy controls. On the other hand, high levels of Faecalibacterium have not yet been clearly associated with particular diseases and conditions, although there are some clinical indications that high levels might be associated with, for example, high intake of carbohydrates or particular digestive problems that may interfere with the digestion of carbohydrates. So this is an article that discusses some of what's known about Faecalibacterium along with other beneficial commensals that are recognized as either keystone species or generally important for the microbial ecosystem and impacts on health. So the title of this article is The Potential of Gut Commensals in Reinforcing Intestinal Barrier Function and Alleviating Inflammation. So right there that summarizes two of the well-known potential relationships of Faecalibacterium to health. It's known to promote the health of the intestinal barrier as well as to reduce inflammation. I'm going to go through a few more take-homes from this particular article, but just wanted to emphasize it here. You'll note at the top that it says PMC free full text. So this is an article that you can download for free from PubMed. And I would encourage you to check this out because this has some great information on a couple of the species that we're going to be talking about today. So with regard to Faecalibacterium, here's one of the quotes from the paper, and they say, F. prausnitzii is one of the most abundant bacterial species inhabiting the human intestine, representing 2 to 15% of the total bacterial load. F. prausnitzii promotes gut health in various ways. For instance, it is involved in the feeding of colonocytes, the maintenance of immune homeostasis, and the strengthening of barrier function. And again, it performs those beneficial functions partly due to the fact that it produces butyrate, and it's one of the main butyrate-producing bacteria. And it turns out that butyrate is actually the main energy source for the colon epithelial cells. So again, it's really critically important for the health of the colon. And in this article, they go on to say, in respect to health, the substantial production of butyrate by F. prausnitzii is of special interest. Butyrate is a major energy source for colonocytes, and it also takes part in immune modulation by inhibiting nuclear factor kappa B, or NF-kappa B, transcription factor activation, consequently reducing inflammation. Because low levels of Faecalibacterium have been associated with a wide range of inflammatory and autoimmune conditions, it can obviously be potentially a benefit to increase levels of Faecalibacterium that are unusually low in particular patients. So there are a number of different well-established ways to potentially do this based on what's known from research and also clinical information. And that would include, in particular, addressing existing gut inflammation as well as any pathogen infections and inflammatory dysbiosis. So all of these have to do with factors that involve inflammation, and inflammation is known to be very detrimental potentially to Faecalibacterium. It doesn't do well under conditions of increased inflammation. So that would be the first thing potentially to identify and address. In addition, there are a number of diet and supplementary approaches that can be helpful to promote Faecalibacterium, and that includes prebiotics, so things like inulin, FOS or fructooligosaccharides, GOS or galacto-oligosaccharides, resistant starch, partially hydrolyzed guar gum, pectin, and then sources of fiber from legumes have also been shown to help promote Faecalibacterium. Another potential category of factors that can promote Faecalibacterium include polyphenols, and those that have been best studied so far in terms of helping to promote Faecalibacterium and other butyrate-producing bacteria would be curcumin, quercetin, resveratrol, and various extracts from berries such as blueberries, cranberry, etc. Lastly, some probiotics have also been shown to potentially promote Faecalibacterium and other butyrate producers. In particular, various Bifidobacterium species can promote butyrate-producing species partly because they're able to potentially cross-feed Faecalibacterium by producing something called acetate. So that's one of the major products of Bifidobacterium species. And acetate is something that Faecalibacterium can convert into butyrate. So again, certain probiotics may be helpful as well in helping to promote Faecalibacterium as well as other butyrate-producing bacteria. The next marker, in addition to Fusobacterium, that has been added to the normal bacterial flora section is Akkermansia muciniphila. So a little bit about Akkermansia. It's also known as a keystone species, and that's partly because it inhabits a particular niche in the mucus layer, the thin mucus layer that is protecting the epithelium in the colon. And so basically what it's doing in this mucus layer is breaking down the mucus polysaccharides, which actually provides sugars then, as well as metabolic products that it generates from those sugars, particularly acetate. It can provide these sugars and products for other bacteria to use as an energy source. And that's particularly important when there may be a lack of fiber and other types of resistant starches and undigestible carbohydrates, such as during fasting, overnight, etc. So that can provide a more steady supply of energy for various types of bacteria that are unable to break down the mucus polysaccharides. And through these actions and interactions with the immune system and the epithelium, Akkermansia can also promote a healthy intestinal barrier, as well as modulate the immune responses. So one thing to note, we just talked about Fusobacterium prausnitzii as a particularly important keystone species, partly because it produces butyrate, which is really important for the overall functioning of the gut ecosystem. And Akkermansia actually can help support Fusobacterium, again, by breaking down these polysaccharides, as well as generating acetate that can support Fusobacterium. So the two of those species actually potentially can work together. So next I'm going to talk a little bit about what's known in terms of imbalances in Akkermansia in the gut. There's actually some associations both with low levels and unusually high levels as well. When it comes to low levels, those have mostly been associated with various metabolic disorders, in particular obesity, diabetes, and metabolic syndrome, whereas high levels have been associated with various neurodegenerative conditions, in particular Parkinson's, as well as multiple sclerosis, and then some new research suggesting that there may be a link between high levels of Akkermansia and Alzheimer's. So again, there may be kind of a double-edged sword here where low levels or high levels may be problematic. Next, I'd like to highlight a couple of recent review articles discussing the role of Akkermansia in human health. And this first article is actually the same article that we just talked about with regard to Fusobacterium prausnitzii. And the title is, once again, The Potential of Gut Commensals in Reinforcing Intestinal Barrier Function and Alleviating Inflammation. And in the article, they say Akkermansia muciniphila is a gram-negative anaerobic mucin-degrading bacterium. By degrading the complex mucin glycoproteins and due to its ability to produce acetate, propionate, and a product called 1,2-propanediol, Akkermansia contributes to the bacterial community by cross-feeding. Akkermansia can metabolically support the growth of butyrate producers by degrading mucin and providing acetate. This is a separate article titled Good Bug, Bad Bug, Breaking Through Microbial Stereotypes, that emphasizes this sort of dichotomy between the association of Akkermansia as being beneficial potentially for metabolic health, but high levels potentially being associated with neurodegenerative disorders. So they say, perhaps the most notable example of the good bug, bad bug phenomenon is the intestinal bacterium Akkermansia muciniphila. It has garnered a great deal of scientific interest due to the consistent observation that intestinal Akkermansia abundance is reduced in a variety of metabolic disorders, including obesity, dyslipidemia, and type 2 diabetes in dozens of animal and human studies. And they go on to say, however, recent findings from the field of neurology provide a cautionary note. Since 2015, 6 studies examining the gut microbiome in Parkinson's disease have reported an increase in Akkermansia abundance in Parkinson's disease patients. Additionally, several emerging studies have reported an increased abundance of Akkermansia in multiple sclerosis. So this article is highlighting Akkermansia as a key example of this phenomenon where a lot of the particularly important or particularly notable microbes that are being identified in the microbiome aren't necessarily strictly, quote, good or bad. It's really context dependent and also dependent to some extent on the relative levels that are in that particular individual. Given that low levels of Akkermansia have been widely shown in various studies to be related to metabolic dysfunction, there's a lot of clinical interest in ways to potentially increase Akkermansia in those particular situations. Some of the best established ways based on research to potentially increase Akkermansia would include polyphenol supplementation, in particular cranberry, grapeseed, red wine, and pomegranate extracts, some of which have actually been shown to substantially increase Akkermansia, and that would include in particular cranberry and grapeseed extracts. Certain prebiotics have also been shown to increase Akkermansia, and that includes inulin as well as fructooligosaccharides. The antimicrobial botanical berberine has been shown to increase Akkermansia, as well as omega-3 fatty acids. And then lastly, intermittent fasting, caloric restriction, and weight loss have all been shown to potentially also increase Akkermansia. And now, based on some of the newer studies that are starting to show that there may be an association between high levels of Akkermansia and various neurodegenerative disorders, there's clinical interest in potentially understanding how Akkermansia levels may be decreased. Unfortunately, to date, based on research, that's not really been well established, something that's not been well studied. However, there is some research that gives some hints as to what may contribute to decreasing Akkermansia levels. So this second bullet point, promoting butyrate-producing bacteria, may inhibit the overgrowth of Akkermansia. That's been shown in at least a couple studies suggesting that the Clostridia group of bacteria, which is the group that contains most of the butyrate-producing bacteria, that those types of bacteria may actually keep Akkermansia from overgrowing. So we talked before about that there's potentially a sort of a mutual relationship, beneficial relationship between Akkermansia and certain butyrate producers, including Faecalibacterium. But it's also been shown that potentially they may help to keep Akkermansia in check. And then lastly, there's some suggestive evidence that indicates that digestive dysfunction might also result in increased Akkermansia levels. So therefore, addressing digestive dysfunction may help to correct those elevated levels as well. All right, so moving on to the third marker that has been added to GMAP. These are the Methanobacteriaceae, and this marker has been added to the opportunistic bacteria section. So a little bit about the Methanobacteriaceae. This is a taxonomic family that's basically the primary group of methane producers in the human gut. And methane producers are otherwise known as Methanogens, and they promote carbohydrate fermentation as well as short-chain fatty acid production by the bacteria in the microbiome by converting the hydrogen that they produce into methane. So if excess hydrogen is being produced by a certain amount of carbohydrate fermentation, that can actually feed back in a negative way on carbohydrate fermentation and slow it down. But by converting this hydrogen to methane, that essentially relieves kind of a bottleneck on the process, and that allows this process to continue forward. So again, they help to promote overall short-chain fatty acid production. And of course, we know short-chain fatty acids have many potentially beneficial effects. So this can be one of the beneficial effects of methane production by the Methanobacteriaceae family. We also know that they tend to thrive in an anaerobic environment, and that they tend to be higher, at least in certain studies that compare healthy versus inflammatory type conditions, healthy populations and those that have inflammatory conditions. So they appear to be potentially inhibited by inflammation in the gut. So in a sense, they may actually function as sort of a marker of sorts for inflammation in the gut. In terms of imbalances, high or low levels of Methanobacteriaceae, low levels have generally been associated potentially with inflammatory bowel diseases, as well as diarrhea-dominant form of IBS. As I mentioned, normal levels are generally just thought to help promote carbohydrate fermentation and the production of short-chain fatty acids, whereas high levels, which is probably the imbalance type that's been most studied, has been significantly linked to constipation in general, and in particular, constipation-dominant IBS. High levels have also been linked to obesity, and then somewhat contradictory, also anorexia. So it appears that either extreme can result in the promotion of high levels of methanogens. And then also interestingly, high methanogens have also been linked to endurance exercise. So there's some really interesting metabolic links between methanogens, especially high methanogens, and various types of conditions or circumstances. So this is a quote from an article that summarizes what's known about methanogens in humans, and the title is Methanogens in Humans Potentially Beneficial or Harmful for Health. And in the article they say methanogens are anaerobic prokaryotes from the domain Archaea. So just a side note here is they are bacteria-like, but they're actually not bacteria themselves. So they in many ways appear to be like bacteria, but they also have some key differences. So they're in a totally different group taxonomically from bacteria, and these again are known as the Archaea. So they say the Archaea that utilize hydrogen to reduce carbon dioxide, acetate, and a variety of methyl compounds into methane. There is convincing evidence that hydrogen removal by methanogens in the gut allows for more effective bacterial fermentation with more efficient production of short-chain fatty acids. And they go on to say recent studies have made links between gut microbiota disturbances and symptoms of IBS, including bloating and constipation. It has been shown that the proportion of methane producers is higher in IBS constipation-dominant patients, i.e. 58% methane producers, compared to IBS diarrhea-dominant patients, where only 28% were methane producers. This is indicative of a possible link between higher levels of methane and slower gut transit times. Just a side note that many other studies have also shown this link, so it appears to be a pretty significant association that when methanogens are higher and levels of methane are particularly higher, that can contribute to slowed motility, potentially in the small intestine as well as in the colon. And this is a pretty helpful figure from that review paper illustrating the various conditions that have been associated with either high or low levels of methanogens. So if you look on the left here, basically these are the conditions that have been established to be associated with a high abundance of methanogens. That again includes obesity and anorexia, so the two sort of ends of the extreme, irritable bowel syndrome, constipation dominant, as well as diverticulosis and periodontitis. So again, just as kind of a side note that methanogens are also found potentially in the oral cavity. So in terms of low abundances, those have been mostly associated with inflammatory bowel disease, as well as irritable bowel syndrome diarrhea dominant, and then some indication that there may be an associated with atherosclerosis as well. So given these various imbalances in what's known in terms of these associations to lower high levels of methanogens, and particularly high levels are probably the imbalance that's most clinically relevant, or it's been most clinically studied. So that's really where the emphasis has been clinically on how to decrease an overgrowth of the methanogens. And some of the ways that have been established to potentially do that include incorporating a low-carb or low-FODMAP type diet, weight loss, especially obviously in relation to obesity. Garlic extract, particularly the component allicin, has been shown to decrease methanogens. Lauric acid, also known as monolaurin, has also been shown to decrease methanogens, as has Lactobacillus reuteri, and then the botanical Moringa oleifera. And then on the other hand, because low levels of methanogens have also been linked to various conditions, there's some interest in understanding from a clinical standpoint how to increase them. Unfortunately, that's not really well established, but based on what we know so far, the following may be helpful in trying to increase methanogen levels if they appear to be unusually low. Certainly we know there's a link between low methanogens and inflammation, and especially in certain inflammatory conditions. So addressing inflammation and providing additional anti-inflammatory support may be helpful. We know that methanogens tend to thrive in an environment where there's a large amount of carbohydrates, especially carbohydrate fermentation occurring. So generally increasing carbohydrates, but in particular increasing fiber content may be helpful. Also, just sort of from a clinical anecdote standpoint, avoiding frequent or extreme dieting may be helpful as well. This is something that I've definitely seen in my own practice, where clients who have been or had a history of frequent or extreme dieting tend to have higher levels of methanogens. And that may be related again to these metabolic associations with various extremes. And we talked about the relationship between high methanogens and obesity as well as anorexia. So that may play into this dynamic of sort of yo-yo dieting leading to elevated methanogens as well. So let's go ahead and move on to the fourth and final marker that has been added to GIMAP, and these are the Fusobacterium species that have been added to the potential autoimmune triggers section. And just kind of a side note that this section is probably best thought of as not only limited to their relationship to autoimmunity, but also all of the species in this section also are generally known to be associated one way or another to inflammation. So it can be generally associated inflammation or more specifically to autoimmunity. And we'll talk about both of those aspects in relation to Fusobacterium. So Fusobacterium species are common commensals typically found in the oral cavity, but they're also found in a fairly high abundance in the upper GI tract. It is known that they may also colonize the lower GI tract, at least in some patients. They play an important role overall in both normal and potentially pathogenic biofilm formation. That's something that's been probably most well studied in terms of the oral cavity and the oral biofilms that they play a role in helping to form. So it stands to reason that they may also be participating in biofilm formation in the gut as well. It's also known that Fusobacterium produce lipopolysaccharide, i.e. LPS, and that may be one way in which they promote inflammation. So Fusobacterium are pretty well established to help promote inflammation in various ways as well. So in terms of imbalances, high levels of Fusobacterium have been associated with certain inflammatory conditions, in particular IBD, colorectal cancer, and then also the autoimmune condition systemic sclerosis. Low levels have not really yet been established with any particular conditions, but there is some evidence suggesting that possibly digestive dysfunction, such as bile acid imbalances, may lead to unusually low levels of Fusobacterium. This is a recent review article titled Fusobacterium nucleatum, Symbiont, Opportunist, and Oncobacterium that provides some good information, particularly on one of the most notable species in that group, which is Fusobacterium nucleatum. So in this article, they say, Fusobacterium species are found in the mouth and other mucosal sites of humans and other animals. Their presence in these healthy tissues suggests that they are natural constituents of the microbiota at these sites. However, as they have been frequently isolated from these same and other tissues in clinical samples during active disease, they are also regarded as opportunistic pathogens. So again, this is kind of that conundrum where they may be in certain circumstances, certain individuals, just part of a healthy, normal microbiome. And then other individuals, based on the context, they may be participating in pathogenic type processes. They go on to say Fusobacterium nucleatum has long been found to cause opportunistic infections and has recently been implicated in colorectal cancer. Fusobacterium has also been isolated from gastrointestinal infections, including appendicitis, and has recently been associated with inflammatory bowel disease. And this next article discusses what's known about the link between Fusobacterium and systemic sclerosis. And the title here is Intestinal Microbiome in Scleroderma, Recent Progress. And in the article, they say recent reports have identified common perturbations in gut microbiota across different systemic sclerosis cohorts. Certain genera may protect against, such as Bacteroides, Clostridium, and Lactobacillus, or conversely exacerbate, such as Fusobacterium and Prevotella, gastrointestinal symptoms in systemic sclerosis. These genera represent potential targets to avert or treat gastrointestinal dysfunction in systemic sclerosis. So because of these associations with Fusobacterium and a range of inflammatory and autoimmune type conditions, there's obviously clinical interest in how to potentially decrease Fusobacterium levels. So far, there's not a lot known, but some research indicates that certain antimicrobial botanicals can help reduce Fusobacterium levels, and that includes berberine and oregano. And then certain polyphenols have also been shown in certain research studies to potentially reduce Fusobacterium levels, and those include quercetin, curcumin, green and black tea extracts, blueberry extract, as well as cinnamon and rosemary. So that brings us to the conclusion of this overview of the new GI map microbial markers. And if you'd like to learn more about the markers, we do have some additional information and resources on our website. So if you go to diagnosticsolutionslab.com, and in the Clinicians section, there is a learning resource section. And in that section, you'll find the GI-MAP interpretive guide as well as the updated GI-MAP white paper that both include information on these new markers. So thank you for listening, and I hope you find that this overview helps you get the most out of our new microbial markers.